Hello everyone, I'm Dehan Cai, a PhD student from the City University of Hong Kong. Today, I will share my work, HaploDMF. It is a tool for viral haplotype reconstruction from long reads. I will introduce HaploDMF by background, method, and result. So let's start from the background. First, what is viral haplotype? We know that the COVID-19 virus has many variants like Alpha, Delta, and Omicron. This is because RNA virus mutates constantly, and these variants have different genetic sequences, but still they belong to the same virus, so we call them viral haplotypes. And the different bases between haplotypes are called single nucleotide variants. Although viral haplotypes have highly similar sequences, some of them can have very different biological properties. For example, the genomes of the Omicron and Delta haplotypes shared more than 99% similarity, but Omicron spreads faster. And people found that a sample may contain multiple haplotypes because of replication of coefficient. So, being able to reconstruct multiple haplotypes in a viral population can help us track the changes of viral genome. One useful way to study viral haplotypes is to sequence them. Suppose there are 3 haplotypes in a sample. The sequencing machine will generate reads from all of them. And given the reads, our goal is to reconstruct the complete genome sequences of all the haplotypes. So, the input is the set of reads, and the output is the genome sequences of all haplotypes. Here, we will use the long reads from the third-generation sequencing technology like nanopore and PacBio. The most important advantage of long reads is that long reads can cover distant ISMBs between haplotypes. However, many long reads datasets have high sequencing error rates. This makes reconstructing similar haplotypes from the noisy data challenging. So, to reconstruct the viral haplotypes from long reads, we designed a tool called HaploDMF, and I will show you how this tool works. This is the workflow of our method. First, we will align the reads to a reference genome, and then we will separate reads into clusters so that each cluster contains reads from the same haplotype. Finally, we will generate the consensus sequences from each cluster and calculate the abundance of haplotypes. In this workflow, the most challenging and important part is how to cluster reads accurately. There are some tools using the overlap consistency to connect reads directly, and they need to determine a cutoff for the overlap consistency. However, it is not trivial to determine the best cutoff because we don't know how similar the haplotypes are in the sample, and the overlap length usually varies. So, instead of using the overlap consistencies to cluster reads directly, we applied a deep learning model called DMF to denote each read with a latent vector. Then we don't need to set the cutoff, we just use the latent vectors for clustering. Because this process optimizes for all the reads at the same time, the latent vectors contain the global information, so it would be better to use the latent vector for clustering. This DMF model was originally applied to a recommender system problem, so I will briefly talk about the recommender system first. Here is a scoring metric for movies. Each row represents the scores from a user. By using the existing scores, we want to predict the scores of movies and recommend them to the users. To predict the score, we can apply this deep metric factorization model to the scoring metric. It takes the row vector of user I and the column vector of movie J as input, and then it represents the user and the movie with two vectors of the same dimension. Then the cosine similarity between these two vectors is used to approximate the score of movie J from user I. Because similar taste users have many identical scores with movies, the learned latent vectors of these users will be similar too. Like here, user 1 and 3 have many identical scores with movies. That means their latent vectors should have the same cosine distance to the latent vectors of movies. As a result, their latent vectors are close. Recall that we actually want to cluster rays. If we can learn similar latent vectors for rays from the same haplotypes, we can then use the latent vectors to do clustering. To achieve this, we will use a base frequency metric. If we replace movie, user, and score with site, read, and base frequency, we will have such a base frequency metric. This metric can be derived from the read alignment easily. Because reads from the same haplotypes tend to have the same nucleotide bases at the corresponding sites, they will have similar frequencies at the corresponding site too. As a result, reads of the same haplotype will have similar latent vectors. Based on this, we will first generate a base frequency metric from the read alignment. Here, we only keep the ISMB size because ISMBs are the fingerprints of haplotypes. And in this frequency metric, we only keep the top two frequencies at each site because the low frequencies are usually caused by sequencing errors. In this loss function, we assign a large weight to the approximation of the low frequency. This is to force the model to pay more attention to the low abundant haplotypes. Because there are other rays connecting to distant rays from the same haplotypes, we can still learn similar vectors for them using this model. However, if there are not enough rays as connections, like the sequencing coverage drop-offs at some regions of the genome, it will be hard to learn similar vectors for distant rays. To deal with this, we force the vector or ISMB size to be closer. Then the distant rays with similar frequency will have similar vectors. Combining loss 1 and loss 2, we learn the latent vectors for rays and ISMB size, and then we use the latent vectors of rays for clustering. We did some experiments with HaploDMF. Next, I will show you some experimental results. We evaluated the performance of HaploDMF and other tools with some metrics like the number of contigs, the error rate, the target genome coverage, and the JSD. Within these metrics, the target genome coverage is the average percentage of haplotype genomes being reconstructed. For example, there are two reconstructed contigs aligned to one of the haplotypes, and the percentage of the two haplotypes are 70% and 0%, so the value of this metric will be 35%. JSD evaluates the distance between the estimated abundance and the real abundance. A small JSD value means the estimation is accurate. Here is the experiment on 2 simulated datasets. There are 10 HCV haplotypes in each dataset. This is the pairwise divergence between haplotypes, and this is the haplotype abundance in the dataset. The red color is HaploDMF. We can see that HaploDMF reconstructed 10 haplotypes and 9 haplotypes with small error rates and small JSD values. HaploDMF only failed to reconstruct the 1% abundant haplotype because the learning model only had a small number of reads from these low abundant haplotypes to train the model. And we found that 1% is the limited abundance for HaploDMF to reconstruct haplotypes. The second simulated experiment has datasets containing 5 HIV haplotypes. This experiment is to investigate the limited divergence for HaploDMF to reconstruct the complete haplotypes, and we found that the limited divergence is about 0.3%. Besides the simulated experiment, we also conducted experiments on real sequencing data. There are 2 HBV haplotypes, 5 HIV haplotypes, and 7 norovirus haplotypes in this dataset, respectively. The HBV and norovirus datasets were sequenced by neuroport technology, and the HIV dataset was sequenced by PEG-by-ORD. The error rates of these 3 datasets are about 10% and 2%. On these datasets, HaploDMF has the best performance. It reconstructed 2 HBV haplotypes, 5 HIV haplotypes, and 6 norovirus haplotypes with the highest target genome coverage and the small error rates. The only haplotype it failed to reconstruct is the norovirus haplotype with the abundance of 1%. Here is the risk alignment depth of 2 HIV haplotypes. We can see that the depth drops off at some regions. Because of this, some tools failed to reconstruct the complete genomes for these 2 haplotypes. For example, RV-Haplo only generated short contigs. However, HaploDMF reconstructed more complete haplotypes. This is because loss tool allows the model to cluster distant reads without enough reads as connection in the middle regions. And here is the visualization of reads in the norovirus dataset. We use the completed frequency metric and the latent betas to visualize reads. By using the latent betas, the color regions have clear boundaries. This shows that the latent betas results in more accurate clusters. In conclusion, we developed a tool called HaploDMF to reconstruct multiple haplotypes from long reads. It takes reads and a reference genome as input and outputs the genomes together with the abundance of haplotypes. Our work was published at Bioinformatics, and the tool can be downloaded on my GitHub page. If you have any questions when using HaploDMF, feel free to drop me an email. And this is the website of our laboratory, and we always welcome collaborations. Thanks for your attention.